All right, welcome again. Today we are going to talk about liver physiology. As you know, the liver is a very vital and important organ in the body, and as a medic, you should know how it functions and the necessities that are in liver physiology, so that you can be able to diagnose any condition like liver cirrhosis, fatty liver disease, alcoholic liver disease, and those hepatobiliary diseases. Okay, let's shift. So we are starting with the overview. So liver physiology is the study of how liver functions to maintain body internal balance, which is the homeostasis in terms of physiology. It stands as the largest internal organ in the body and performs many major functions such as detoxification and metabolism. So blood supply at the liver, it's very special and unique because it has a dual blood supply. It's supplied by hepatic artery, which supplies oxygen rich blood, and also from the portal vein, which supplies nutrients rich blood. So blood flows through sinusoids where hepatocytes exchange nutrients, toxins, and metabolic products before blood drains to hepatic veins and inferior vena cava. Sinusoids are just like blood vessels, capillaries that allows exchange and is always used especially for exchange of substances with the hepatocytes. Okay, let's continue. We'll start with carbohydrate metabolism, where we'll talk about glycogenesis, which is the conversion of glucose into glycogen for storage, as you know. Then glycogenolysis, which is the conversion of glycogen into glucose for storage. We have gluconeogenesis, which is the production of glucose from amino acids, lactate, and glycerol during fasting. All this helps in maintenance of blood sugar levels in the body. Number two, we have protein and lipid metabolism. In the protein metabolism, we'll talk about synthesis of plasma proteins like albumin, then deamination or breakdown of amino acids into proteins. We have also conversion of toxic ammonia into urea reducible. We also have lipid metabolism. As you know, lipids, fats and oils, synthesizes cholesterol, oxidizes fatty acids into energy, replaces lipoproteins, majorly the high density lipoprotein and very low density lipoprotein. Number three, we have bile formation. As you know, bile is produced in the liver and the amount is always 600 to 1000 ml per day. So bile functions, we have emulsification, aids in digestion and absorption of fat soluble vitamins, and excretes bilirubin, toxins, drugs, and cholesterol also. Let's see the bile flow, which starts at the hepatocytes, the liver cells, and ascends to the bile canaliculi, where it's directed to the bile ducts and common hepatic duct takes it through to the gallbladder for storage. Okay, let's look at bilirubin metabolism. As you know, bilirubin is a liver function text, and it starts with red blood cells breakdown, which contains the hemoglobin and reduced cells. So hemoglobin is converted into unconjugated bilirubin first, where the liver comes in and turns the unconjugated bilirubin into conjugated with the affection of glucuronic acid. Then the conjugated bilirubin secreted into the bile and converted into urobilin, which produces with yellow urine, and stacobilin, which presents also with brown stool. Let's look at detoxification, which is the removal of toxic substances from the body. Drugs, alcohol, hormones, toxins, these are the examples of detoxified agents. We also have immune function of the liver. It contains Kupffer cells. These are just special macrophages that are responsible for removal of bacteria from portal blood and destroy all red blood cells, also regulate immune responses in the body. Let's look at endocrine functions, the hormones now. And liver activates vitamin D. Liver also produces insulin growth factor 1, majorly for growth hormone. It produces angiotensinogen. Remember the RAS system for BP management. Then it produces thrombopoietin for platelet production. It also metabolizes insulin and steroid hormones. It also has coagulation nature, where it synthesizes most of clotting factors. Examples are factor 1 fibrinogen, factor 2 prothrombin, factor 5, 7, 9, 10, 11, you read antithrombin also, protein C and S. You'll read those ones, those factors, coagulation. Also, the liver has a regenerative character, which comes in after injury or surgical removal using hepatocytes, which can proliferate to restore liver mass, provide enough healthy tissue remains. That's a picture of the liver, as you had already seen in the first slide. Thank you and subscribe.